Good morning, and thank you for tuning to our fourth webinar of our eDNA webinar series. Our topic today is pushing the boundaries of environmental DNA. My name is Mario Thomas, co-founder and CEO of Precision Biomonitoring, and your host today. Joining me for this webinar is Dr. Robert Hanner, Associate Professor in the Department of Integrative Biology at University of Guelph, and member of the Biodiversity Institute of Ontario and the Center for Biodiversity Genomics. But is also a co-founder and chair of the Scientific Advisory Board of our company, Precision Biomonitoring. If during the seminar you lose a sound or image, you should refresh your screen to solve the issue. Also, make sure you close all other programs and apps that you may have opened on your device. As we all know, environmental DNA or eDNA is a relatively new science that has started to revolutionize industry's approaches to biomonitoring. In the previous three webinars, we have discussed eDNA as a whole, the platform that Precision Biomonitoring has developed for eDNA qPCR. We have developed a webinar on survey design, conducting surveys you can trust. We have also discussed the ecology of E Dna, how it behaves in aquatic systems and in varying environmental conditions. In addition, we have discussed how we have a strong understanding of this ecology of eDNA and how it can help eDNA science. So if you've missed any of the previous webinars, they are always available for viewing on our website. What we are discussing with you today is pushing the boundaries of eDNA, science and technologies. I wish to express our sincere thanks to Stantec, ACOM, Azimuth, Parks Canada, Ecometrics, Smithroot, and Baumeme, and all the collaborators who have helped us build and validate this exceptional platform, initially developed at University of Guelph in Bob's lab and launched commercially last year. Our agenda for this morning is a 30-minute presentation by Bob, followed by a question period of 25 minutes. At this point, I would like to invite Bob Hanner to talk about his views on pushing the boundaries of eDNA. Bob.
Thank you, Mario. Good morning, everyone. Thanks for tuning in. We'll move to the next slide. So I wanted to start with a brief history of DNA analysis in forensics. DNA has become a powerful tool to law enforcement and regulators, beginning first with the identification of nuclein, or what we now know as DNA, by Friedrich Miescher, who identified this in 1869. Then in, of course, 1953, we had Watson and Crick, who discovered the 3D structure of DNA. By 1984, we had Sir Alec Jeffreys proposing DNA profiling when he found that individuals could be differentiated by their DNA sequences and structural variants in their DNA. In 1987, forensic DNA analysis was first used in a criminal case in the UK. And then again in 1987, that same year, evidence sent another person to prison for raping a woman in the United States. And then by 1995, a UK national DNA database was established, and the Scientific Working Group on DNA Methods, or SWGDM, in the United States began to develop human DNA forensics into a powerful tool. But the application in non-human space lagged behind. We didn't start to see routine use of DNA in prosecuting wildlife crimes until the last decade. Next slide, please. This has largely been as a result of the DNA barcoding initiative, where we use short gene sequences to identify species. By genetically profiling known species, we can use this as a lookup table to sequence an unknown and compare its sequence to a database and infer an identification using a variety of short standardized marker genes for use in different taxa. Cytochrome oxidase 1 typically used in animals, ITS in fungi, and a suite of chloroplast markers in plants. This has really allowed us to start to take DNA-based identification to new levels. Next slide, please. The barcoding process is fairly straightforward. We do it routinely in molecular biology labs at universities around the world, where we start with the sample, we take a bit of tissue from that specimen, we extract DNA, PCR amplify the marker gene of interest, sequence it, compare that sequence to a database, and infer an identification. Next slide. New technology, high-throughput sequencing, is allowing us to do massively parallel sequencing of barcodes from mixtures, in essence giving us a sort of species scan. So we've been able to take bulk samples of trapped insects and do what we call metabarcoding using high-throughput sequencing approaches to try and identify what's in a mixture. Next slide. Similarly, we can also exploit the species-specific patterns of variation in a barcode library to make PCR primers and molecular probes that will target that species uniquely. We use this in a technique called real-time PCR. This has some advantages and some limitations. It will allow us to identify a target in a complex mixture, but it doesn't allow us to identify more than one or a few targets at a time. However, the real benefits are in cases where we're dealing with things that are time sensitive, or worried about the degradation of our samples. It allows us to actually do this kind of testing in the field because we don't rely on DNA sequencers and a database. The bioinformatics is built into the assay at the front end. So it's taking a couple of steps off the barcoding approach and allows us to do validation on site. Next slide. These two methods have been used for environmental DNA detection. The purpose of today's seminar is to talk about how we're using these DNA methods to detect DNA that's been shed into the environment. In this case, typically we're working with aquatic species that are sloughing off skin cells or epithelial cells or gametes into the environment, and they can be detected by eDNA. And academics have been using this tool for the last several years. As you can see here, the number of publications has been growing exponentially to detect environmental DNA in various ways. Next slide. These eDNA workflows can follow either of the two technology pathways that I described previously. We can infer the presence of a target species using qPCR, or we can attempt to look at the composition of a community through metabarcoding. And there are targeted approaches that typically use quantitative PCR, although droplet digital PCR is emerging, and we'll come back to that later in the seminar. Similarly, with high-throughput sequencing, there are a number of different platforms that have been developed and put forth, each with their own idiosyncrasies. So two main pathways depending on your question. Next slide. So what is your research question? Are you simply looking to monitor presence absence? Are you looking at habitat preference of movements of organisms in and out of different habitats? Or are you trying to understand a community or an ecosystem? What do you really want to detect? A target species or a specific set of species? Or are you trying to take attendance of lots of species, which would dictate more of a metabarcoding approach? Other things that we need to consider as we employ eDNA tools is the species common or rare? Is it invasive, endangered? Next slide. When we're trying to use environmental DNA as a tool for detecting aquatic species, we need to think about the natural history of the organisms that we seek to detect. Where and when is it found? When is the target species expected to be most abundant? During breeding season, migration? What habitat types are they using? We need to consider these issues as well as their behaviors. Do they have preferred habitats where we should be looking for them? that would inform our sampling design strategy, where they feed or seek shelter. Also, are there seasonal variances in their abundance? Do they migrate, spawn, or overwinter in different habitats? All of these aspects of natural history must be considered as we think about how to properly detect species using environmental DNA. Next slide. So this leads to sampling considerations. How and how much to sample? How many samples to take is a key question, and it can depend on the species and their life history, as we've just discussed. For example, maybe 10 biological samples may be sufficient for a pond or a terrestrial habitat, but perhaps 300 samples may be needed for a large riverine study, depending on the scope of your question. We also have to consider environmental characteristics that may affect eDNA concentrations. Temperature, pH, UV radiation, flow, a variety of physiochemical parameters can influence the state of eDNA and its persistence in the environment. Ideally, we should be taking eDNA samples prior to other types of samples. These methods are so sensitive that they can be prone to contamination if we're moving physical nets and boats and gear from one site to the other. So you would ideally want to make sure that you're not introducing any kind of contamination, and if you are, that you're bleaching it to remove any cross-contamination, and that we need to employ proper controls at all stages in our experimental design, from sampling and extraction through to the PCR tools that we use to identify the presence of a species in an environment. Next slide. In my lab, we've been working mainly with targeted approaches. Although we have done some metabarcoding, for more routine applied use, we feel like the qPCR approach is a bit more reliable when we're looking for a specific target. We typically follow the MICE guidelines, minimum information about a quantitative PCR experiment, as a guideline for developing and reporting of our assays, and we include internal positive controls that allow us to have a very sensitive measure of things like PCR inhibition. Furthermore, we're fortunate to have an ISO accredited laboratory methods validation group at our laboratory services division who can perform third-party validation on all of the assays that we've developed. We're targeting short fragments because eDNA degrades very quickly, so we must look for small fragments in the environment, and we calculate our limits of detection and quantification, the efficiency of our reactions, and this has informed the methods that Precision Biomonitoring has used as it's developed a toolbox for investigating environmental DNA occurrence of buried aquatic species at risk, invasive alien species, or other valued ecosystem component species. Next slide. I've championed this PCR-based approach because it allows us to bring the lab into the field. We've been working with portable and integrated water collection and filtration systems, typically using the Andy from Smith Root. We then use DNA extraction kits from Biomeme to extract the DNA in the field. And we can even do qPCR on portable qPCR platforms in the field as part of our pilot studies to assess the performance of our assays and help inform larger experimental designs based on site-specific environmental parameters. I'm optimistic about this approach because taking the lab into the field not only saves time, but it minimizes the signal degradation from collecting a water sample and trying to transport back to a lab. Many of the sites that we're working at are remote, and it's simply not practical to be able to get these water samples back to a laboratory for filtration in a timely manner. So we know that the eDNA can continue to degrade in those water samples if they're not properly filtered for analysis. We've also seen issues certainly in academic labs where we've been generating a lot of PCR products in our routine DNA barcoding studies that can contaminate our lab space. And actually moving out into the field and outside of existing PCR labs has helped to minimize some of our contamination risk. Next slide. So I want to come back to and really stress what I think are some of the factors that must be considered during an eDNA analysis. There are many factors that can complicate or confound an analysis that we need to be mindful of. I mentioned contamination and signal degradation. We're trying to detect tiny amounts of DNA, and careful collection and preparation of samples for analysis are vital to prevent unintentional contamination or degradation of the sample. We also have to think about things like primary occurrence versus secondary transfer. It is possible for aquatic organisms to be moved around, or even their eDNA to be moved around through predation or other sources. So it is possible that you can see minute traces of eDNA, even though the organism may not be in that environment. And proper experimental design can help you elucidate whether it's primary occurrence or secondary transfer. Also, when we're developing these assays, we have to think about evolutionary relationships, both within and between species. If an assay is validated on a very narrow set of specimens that don't represent the standing genetic variation of a species across its range, it may miss some haplotypes of a species in a population that was not originally included in the assay design. Or closely related species may not have diverged enough to tell them apart. If there's still hybridization and we're using a mitochondrial marker that is maternally inherited, we need to be mindful that maybe our resolution can only get us down to a pair of very closely related species. So we have to think very carefully about the evolutionary history of the species that we're looking at. And again, this comes back to this issue of pilot studies. If you're using an assay in a novel area where it hasn't been tested before, ideally you need to obtain some tissues of that target species to ensure that that assay will work on that population in that region. Next slide, please. Additional factors that we need to consider are things like complex profiles. Samples where the DNA levels are extremely low or degraded, where there are PCR inhibitors present, or even extreme amounts of biological activity. We've seen that can complicate analysis and raise uncertainty. Another issue that we face in dealing with environmental DNA detection is the software. Scientists use computer programs to assist them in evaluating the DNA profile similarity of different samples and calculating match statistics. But different software packages are suited to different sample types and complexities. So we must be mindful of this in choosing our appropriate tools. And this is really important when we think about metabarcoding, where different pipelines have been developed primarily for microbial research, and we're trying to extend and apply them to other taxa. But it also holds true for how we design our qPCR primers and probes as well. Not all software packages are created equally. Next slide. So this leads me to the denouement in this story, what I'm calling the Unholy Trinity. There are three primary areas of uncertainty that we have to deal with when we are attempting to use environmental DNA as a tool to detect aquatic or other species. So in a landmark paper by Barner and Terns, they coined this term the Ecology of E Dna. They pointed out that a whole new field of study has emerged around understanding the origin, state, transport, and fate of eDNA. Origins refer to things like shedding rates. How much eDNA would we expect an organism or a population of organisms to put into the environment? The state of eDNA is another interesting and not fully resolved question. Does it exist primarily as cellular debris? Is it naked DNA that's released from broken cells? Some combination of the two. This state of the DNA is important in terms of thinking about how we detect it. Also, its transport. How far will it move? Does it diffuse? Does it settle out? And ultimately, its fate. What are the factors that affect its degradation? Is it microbial activity that consumes and recycles these phosphate-rich DNA molecules? Is it UV light breaking them down, or other enzymes that are in aquatic systems? We still have work to do to fully characterize the factors that are impacting the origin, state, transport, and fate of eDNA. But much work is being done in this space. On a more pragmatic level, we have issues around the methodology of detection. How many samples and what volume to take? And how do we collect or concentrate that sample and filter or extract and preserve it or extract it? What detection method are we using? Are we taking a qPCR based approach, or are we looking at a platform for metabarcoding and the varied hardware and software requirements that go with that? What kinds of primer biases might impact our study? There are many different aspects of methodological concern when we think about how best to try and detect environmental DNA. And lastly, we have the environmental conditions, both biological and physiochemical. We know that there are some compounds in aquatic systems that can be inhibitory to PCR. Some compounds can actually bind DNA. So we have binders in the system. And in extreme cases, we can have lots of other DNA from non-target organisms in very microbially active systems where there's too much extraneous DNA competing with your target. So we have to think about binders, competitors, inhibitors, but we also have to think about things like flow rates and water body volume, pH, substrate, temperature, UV exposure. All of these factors of ecology, methodology, and environmental condition play off of each other, such that optimizing a particular approach for a particular target species in a particular environment quite often requires some preliminary investigation or pilot study to make sure that you have proper detection probability in that particular environment for that species. My position is that it's still too early for a generalized prescriptive approach to detect all taxa in all systems. We're still really trying to understand the boundary conditions that govern how we can detect environmental DNA. Next slide. But the good news is it's working. We're seeing the studies every day coming out that have been able to detect eDNA found in glaciers and snow and aquatic systems. But as I said, we're still trying to get a better grasp on things like the boundary conditions that govern how we can reliably detect that signal. Next slide. So one of the things that I wanted to begin to close with is thinking a bit about the future. Where will we be in another 3 to 5 years? This is a rapidly evolving field of research, and typically we've seen people attempting to just take a water sample, take it back to a lab and filter it. But I think we're going to see the emergence of use of more autosamplers. Next slide. In a paper that just came out at the end of 2018, they used the same apparatus to look at composite versus grab samples in a metabarcoding study that was spanned both upstream and downstream of a fish hatchery. And given the potentially high variance in aquatic eDNA among replicate grab samples, compositing smaller water volumes collected over a period of time may be more effective for some applications than a simple grab and go approach. And it was interesting, in their study, they found that rarefaction curves suggested faster taxon accumulation for composite samples, and estimated richness was higher for composite samples as than for a set of grab samples. So we can think about compositing things through time, but also some of the work that's been done here at Precision Biomonitoring, and also through space in terms of collecting aquatic eDNA along a transect as opposed to a static grab sample. These kinds of things may enhance our probabilities of detection. And interestingly, coming back to that interplay of the Unholy Trinity, they found that the salmonid eDNA dropped out substantially faster than the Coregonus or Whitefish eDNA between the two sites, regardless of sampling method, suggesting that differential settling affects the estimation of relative abundance. So again, what's going on here? Does it have something to do with the size of the cells that are being shed from the different species? Why would we see a taxon-specific difference in settling rate? Studies like these are illuminating new aspects of this ecology of E Dna. So it's an exciting time to be doing research in this space. Next slide. I think as we move forward, we're also going to see more accurate quantification. Again, this is one of the reasons that I was attracted to a quantitative PCR approach for a target species, as we can get relative signal intensity using our qPCR assays. That doesn't necessarily correlate to organismal abundance, but it might. And some studies suggest that it can. And I think we'll be able to move towards more accurate quantification of particularly very rare eDNAs using some new tools like droplet digital PCR that are starting to become more routine. Next slide. I also think we're going to see more hydrodynamic modeling informing our sampling strategies. My colleague Chris Wilson at Ontario Ministry of Natural Resources has done some really interesting work using sonar to look at where in the water column the fish are to figure out what depth to sample. Last week, I was invited to speak at the Great Lakes Institute for Environmental Research, and Dan Heath and folks there have been doing some interesting work and looking at hydrodynamic modeling and large transect surveys of eDNA to see how eDNA behaves in these systems that are dynamic and flowing. Again, we will learn a lot about how to sample through combining these sorts of approaches. Next slide. I think we'll also begin to see better tools for data visualization. This is a web shot of an NVIR data set, but there's no reason we can't imagine as large research programs get underway and lots of data become mobilized around eDNA, that we can start to do this kind of heat mapping of eDNA for different species, looking at invasion fronts in near real time. So just to begin to wrap up, next slide. In October of 2018, last fall, we held a national workshop at the University of Guelph that brought in many members of the community from primary industry and the environmental consulting firms that serve them, through to regulators, including folks from DFO and Ontario Ministry of Natural Resources and others, as well as a variety of academics to talk about where we are in the state of eDNA and its uptake. Obviously, both active, targeted, and more passive metabarcoding approaches require significant R&D for primer development. Standards and best practices are emerging, but as we've discussed, a significant heterogeneity of methods remain and may be needed for different applications. One thing that we saw that was a bit troubling is that metadata on these studies is being rather inconsistently reported, and this is inhibiting our ability to really fully understand aspects of the ecology of eDNA and those environmental conditions that impact it. So we have unanswered questions around the ecology of eDNA. We need to think more about the implementation of eDNA in the context of habitat occupancy modeling. We need a data archival strategy for this occurrence data. And we decided that really it was time to coalesce a community of practice here in Canada to begin to drive forward this promising new technology. Last slide. Which leads me to point out that we will be revisiting the Pisces in late May of 2020 back at the University of Guelph. We're establishing a listserv for those who would be interested in keeping abreast of the meeting. Maybe you have results you'd like to present there or just learn more about the tools and technologies being employed, but we hope to see you there. Thank you very much.
Thank you very much, Bob, for sharing with us your knowledge and insights about the boundaries of environmental DNA and how we can, through creativity and rigor, push them to enhance the value of this promising technology. As we have seen and heard, applications of DNA can be reliable if and only if conducted with statistically sound survey designs, with a proper pilot study, with proven sampling methods and sample preparation, and with accurate and validated qPCR tests. It is a science and a process, a lot more sophisticated process than a simple grab a sample and go ship to the lab. So before we turn it to you to ask any question, I would like to explain how to do this. At the bottom of your screen, you will see questions. And if you click on questions, you'll be able to type in your question, which in turn I will read aloud for Bob to answer. So don't be shy. Bob answers all questions. I know this for a fact. So type in your questions and we will address them. So while you're thinking about the questions to ask our co-host today, I have one question for you, Bob. Do you see the day where eDNA could be used to determine the abundance of a specific species in a water body?
That's a really good question. It's one that occupies the minds of many of us working in this field. We've certainly seen a number of publications suggesting that signal strength from things like targeted detection in qPCR correlates with abundance. We've seen cases where that relationship isn't so strong. Some of our work has shown that when we include an IPC or internal positive control and can actually normalize for inhibition, what didn't appear to be a correlation between an inverse correlation between CQ value of qPCR and abundance actually begins to emerge when you correct for that inhibition. And we've recently seen that in some work done at the Detour mine site around Burbot, where this is, again, underscores the importance of being able to measure inhibition and correct for it. And so, again, teasing out some of these complicating factors, I think we will get closer to that. We can certainly imagine scenarios that would complicate that. But there have been other studies by U.S. Fish and Wildlife in Alaska where they've used some of the salmonid primers and probes our lab has developed and paired eDNA signal for targeted detection of returning salmon spawning at weirs, and they had camera traps on a number of these weirs so that they had physical counts of the fish coming over the weir, and they also had their eDNA signal that they collected and found a very strong correlation between abundance and eDNA signal.
Thank you very much, Bob, for answering this question. Once again, if you look at below the screen for the presentation, you see a button question. You can just click on it and type in your questions so that Bob can address them. So far, we have not a question, but I do have many questions for you, Bob.
You always do, Mike.
Yeah, I know. That's what makes us a good team. So we talked about qPCR, its benefit of sensitivity, metabarcoding. It has the benefit of looking for community. So what do you think, for what type of applications would you prefer to use metabarcoding than qPCR more specifically?
Well, in some of my academic projects with graduate student theses where we're interested in basic biological questions around community composition, we find eDNA metabarcoding to be an interesting tool. And we've certainly used metabarcoding in things like gut content analyses. For more management-focused decisions, I feel like there are fewer variables that can go awry with a targeted qPCR approach. So that's, as you know, most of the work we've been doing with funding from INSERC and Ontario Centers of Excellence has been to develop primers and probes for species at risk, potentially invasive alien species. I think it's clear, though, when you don't know what you're looking for and trying to do a large kind of system scan, then you have no recourse but to use a metabarcoding approach. But the time delays in terms of collecting, analyzing that data don't give us quite the same near real-time information that we get out of the targeted qPCR-based approaches.
Thank you. So we have a question from the audience here. So let me read the question. Is there any way to reduce the risk of false negatives associated with genetically distant populations of the same species. For example, review the DNA bank to provide higher flexibility?
So that's a really good question. We're always dealing with limited knowledge about the standing genetic variation within a species. So a good example of that is work that a former PhD student, Lauren Overdyke, had done in the lab, building an assay for Lake Whitefish. We were interested in Lake Ontario. We managed to acquire hundreds of specimens from dozens of known spawning sites around Ontario. We DNA barcoded them, so we had a good idea of the amount of standing genetic variation in Lake Ontario whitefish. There wasn't a lot. We could make a very robust assay for it, but would it work on whitefish in, say, Great Slave Lake? I don't know. That's partly why I would say if you're going to extend an assay into a novel habitat, if we don't have any representation of, say, the whitefish from that population, it would be nice to barcode them and make sure that your assay is going to work in that range. We can't assume that it will, because we do know that phylogeographic processes drive some minor genetic differences within species across their range. So ideally, we would like to archive the actual the sequence alignments that we use to build our primers and probes, which is what we do in my lab. And so as new data comes into the database, as the questioner points out, we can go back and bring any new data into our alignment and look to see if that's going to be a potential problem for our assay. Sadly, right now it's not that common for people to include their sequence alignments with publications. It's part of the reason why I think not all published assays are fit for eDNA purposes. They may have only been designed for endpoint PCR and not qPCR, or they may have been designed around a very limited subsampling of the standing genetic variation of the target species. So it's an area that we can continue to revisit if we do good science.
Thank you, Bob. We have another question here, which is, are there quality control issues that are foreseeable with field on-site technology compared to laboratory technologies?
Absolutely. We have to perform proper quality control no matter how we choose to analyze samples. And while we've done a lot of work in the field with great success, sometimes it is more convenient if you have a really large volume of samples to be working back in a laboratory, where we may need to employ cleanup steps to remove inhibitors that aren't easy to employ in the field. So we have to have proper controls to ensure that we're not contaminating samples, that our reagents are working. So we need both positive and negative controls at each step of the way, from reagent blanks through to sample blanks, and ideally also testing some known targeted sites in the field to make sure that your assay is, again, working. It's one thing to look in a site where you hope to find but may not find a species at risk, but we'd like to also pair that with, is there a known site that we can demonstrate that the protocol is working, whether it's lab or field-based.
Mm-hmm. And I may add to this, as you know, we at Precision Biomonitoring, because of these potential issues, have developed very tight and rigorous standard operating procedures that go over and beyond what might be expected. So it's that kind of research in issues that enable us, when we transfer this to the field, to commercial use, we know what to look for and can devise appropriate quality control measures.
And that's important. And I would say equally as important, what we're really learning is, because it's appearing as though much of the eDNA exists in clumped cellular debris rather than naked DNA, it's patchily distributed in the environment. So there's this issue of controls. We want to minimize false positives and false negatives, but that sampling design is going to be absolutely critical in terms of, you can also get a false negative from just insufficient sampling. And that's another area that we've put a lot of work into, is thinking about the amount of spatiotemporal replication required to help maximize the probability of detection around the needs of each particular study.
We have another question from Austin Thomas, our partner. Austin, who is asking, what do you recommend, Bob, to new practitioners for structuring a pilot study? Cage trials, covariates, etc.
That's a really good question. And it's going to, my son hates this. Every time he asks me a question, I tend to answer with "it depends." But Austin, I'm sorry, I'm going to do this to you as well. And you know this yourself. I think that's why you've asked such an excellent lead-in question. These kinds of pilot studies, certainly you want to look to see if your internal positive control is showing any signs of inhibition. So that's one of the first things we're looking for in a pilot study is, are there environmental parameters that are likely to inhibit PCR? Then, of course, if you can begin to do a longer term study and looking at covariates and cage studies, that's obviously very useful to do. And we've certainly done that around a project that was a collaboration with Precision Biomonitoring to develop an assay for wavy rayed lamp mussel here, in a species at risk here in Ontario, where we did some cage trials trying to assess how far downstream we could reliably detect and what kinds of population densities are needed to produce a signal. And interestingly, some of the things that we've come back to in terms of natural history seems like amphibians and fishes that are mobile, moving around the environment, shedding quite a bit of eDNA through their slime layers, and active metabolism, defecating, shedding epithelial cells from their intestinal linings. They seem to be pretty easy targets for detection. Some of the work that we've moved into in the more of the benthic specimens suggests that simply grabbing a sample of water from the surface is not going to give you significant power to detect that species. So again, coming back to life history, we're now beginning to understand the importance of spatial sampling, sampling at depth. So yeah, I mean, that's part of where I say with that unholy trinity, there's still quite a bit of uncertainty to tease out. But again, I come at this very skeptical with every new test and study we design, and I'm pleasantly surprised to see the power of the tool. It's typically outperforming conventional methods, but still is, like any sampling method, is imperfect.
Thank you, Bob. The next question is, good morning. I'm looking at starting an eDNA project in Alberta. What would you say is the best resource to reference in order to establish a pilot study within the laboratory, i.e. aquaria and field cage experiments, to ensure best practice and consistency?
The best resource to reference. That's tough. This is a rapidly expanding literature, but I think some of the seminal papers that you absolutely must be referencing come from Karen Goldberg, particularly your 2016 paper, but there are several others as well that have started to lay out some of the best practices that we're looking for. And also look for papers coming from the first Pisces meeting. We'll have a special issue in Louis Bernatchez' new journal, Environmental DNA, coming out later this fall, where we're talking about some of these issues. But yeah, I would say some of my favorite references are from Karen Goldberg, but also Taylor Wilcox. There are many other very talented people publishing in this space, but the seminal papers, I think, certainly start with Goldberg.
Thank you very much, Bob. Heather, we have your email address. If you want, we can email you a list of references to look at on the topic. Next, a question that I'll take. The cost of eDNA method versus regular method for ecology survey applications in a real world project. I'll take this question because we do this every day, Henry. And what we have done on a number of scenarios for surveys, we have pulled together quotes from environmental consulting firms. They knew that we were going to use this for comparing to eDNA. And we looked at 6 different scenarios. We took an average of the cost that was going to be quoted. And then we detailed what was entailed in an eDNA survey to answer the same question. Depending on the scenario, the cost saving of using eDNA was a minimum of 25% and could be as high as 68%. So the conclusion of this is that for some applications, eDNA is a better choice than conventional methods because of its accuracy. But it can also save significant costs that really add up.
And I would just like to add to that, beyond the actual dollar cost, the fact that this is a passive approach that isn't imparting any stress or mortality on the species that we're actually trying to monitor is a significant value as well. We've certainly done some paired studies with electrofishing and eDNA detection and found greater sensitivity from the eDNA than the electrofishing. And in this case, we didn't have to have anyone, potentially even carrying this kind of equipment. Obviously, best practices for electrofishing are going to require 2 people on site. Whereas when you're sampling with an Andy, if you can have 2 people on site, I think that's always a good idea, but simply slurping a bit of water from the bank is probably safer for your staff, and also less risk of moving not only contaminants, but invasive alien species around too. So I think there are lots of reasons why I find this to be a very exciting tool in addition to the cost savings.
Thank you, Bob. Next question. If the rate of DNA degradation differs from one species to another, do you think that developing standardized eDNA protocols for sampling and analysis should be species specific?
Yes, I do. And this is an area, again, where we're only scratching, seeing the tip of the iceberg in terms of doing some of these kinds of comparative studies. Mario had asked me to keep my presentation to 30 minutes, and we academics will pontificate mercilessly if you let us, but some of the work that we're doing in the Limnotron at the University of Guelph's Biodiversity Institute of Ontario has been looking at some of these kinds of issues. And one of them, interestingly, just looking at putting cellular debris into a tank versus naked DNA and looking at the differences in persistence times. Even within species, I think we can have some interesting questions. Are we talking purely about degradation of eDNA sloughed off from epithelial cells versus persistence of eDNA at spawning time when gametes are released? There's a lot of science that we need to do here to really nail down at the species level exactly what's going on.
Yeah. On this, what we have come to realize in working with the HANA Lab is that on one hand, we need a standard tool, standard method to design a survey. We need to customize the sampling strategy. So this is, a clear case where one size fits all will not work. Very simply, even for the same species, a different study calls for tailoring the survey design and the sampling strategy to maximize the probability of the DNA detection. And this is so important that we have taken the step. We now have a mathematician on staff, and as of next week, one of Bob's master's students in bioinformatics is joining us. We are developing software tools, algorithms, where we can put a ton of metadata to obtain a recommended survey design, attaching to it a probability of detection, so that for a given species, for a given site, we will soon be able to say, this is the recommended survey for that we have, and this is the power of that study, rather than using best estimate and trying to use a standard approach that will not provide optimal statistical value. Thank you very much for this question, because as you can see, I'm very passionate about this aspect, or all aspects, but this aspect in particular. Another question here from someone you know well, Doug Bradley from Limnotech. Doug is asking, what do you see as attractive in the use of digital droplet techniques? And for the interest or the attractiveness of this, are you seeing decreasing the cost of DD machines over time?
Well, that's a great question. Thank you, Doug. I like the droplet digital platform because it can get to very low limits of detection, a little bit better than we typically get for qPCR, particularly for some of the portable instruments that are good in the field, but not necessarily the most sensitive. So in cases where you want that absolute highest power, best probability of detecting, we see that ddPCR gets better limits of detection, but it also seems to be able to deal with some aspects of inhibition. By parsing your sample out into 20,000 different droplets, you're diluting out inhibitors. So that's another thing that I see attractive about ddPCR. But your point about price is a good one. I mean, these instruments are still prohibitively expensive for much more than R&D at this point. We don't use them routinely. I'm not sure we need to for a lot of the applications where the eDNA that we're looking for is in enough abundance that level of sensitivity isn't required. So again, coming back to that idea of a pilot study, there may be cases where you want that level of sensitivity, but a lot of the work we're doing right now doesn't need it. But I do welcome cheaper instruments. And we've seen that happen in sequencing space through time. So I think there's no reason to expect that we won't see a similar decline in price of ddPCR machines in the years ahead.
Thank you very much, Bob. I think this is the last question for this session. Very timely. It's almost 11:55. So we are reaching the end of this webinar. If after this webinar you wish to watch it again, or you want to send it to other people to watch, you will be sent very shortly an email with a link to see a replay of this. I know that one of you had asked a question if it would be possible to have a copy of the webinar. So this is how you can get it. It will also be posted on our company's website, precisionbiomonitoring.com. So I thank you very much for your attention, your interest, and sharing your passion for developing and adapting new technologies that can make everyone's life better. So thank you very much for your attention, and we are going to close the session at this time. So thank you again. Thank you, Bob.
My pleasure. Thank you. And good to see so many of you.